Thank you guys for all coming to my talk on the Trail of Snails, even I get mixed up with it, of the Upper Delaware, plus an unexpected mystery. This project was a collaboration between the National Park Service team of the Upper Delaware River Scenic and Recreation River and the macroinvertebrate section of the Patrick Center at the Academy of Natural Sciences of Drexel. All right. So first I wanted to introduce the Upper Delaware Scenic and Recreation River and to show you all where the park is located. The Upper Delaware is about 70 miles of the Delaware River that forms the boundary between New York and Pennsylvania. One of the unique things about the Upper Delaware River is that there are no dams downriver to the ocean. So there are an array of migratory fish and other life that those populations support all the way up to the top of the river. So you can see where we are in Reading. And so it's about a 3 hour trip to get up there. So here's a closer look at the park. The Upper Delaware is part of the National Wild and Scenic River System, which are there to preserve natural and cultural and recreational values. There have been many research projects over the years, but none of them have focused on snails. So that brings us to the question of, well, why snails? Snails are an important part of the freshwater ecosystem. They play a significant role in nutrient cycling, and they are a food source for many different waterfowl, fish, and some mammals. In the summertime, up at the park, snails can be very abundant. As our friends at the National Park Service noticed, there were a bunch of snails along the edges of the river. And when they went to look up what kind of snails were there and what the snail community was like, they had more questions than answers. Compared to all the other taxa groups in this region, snails were relatively understudied. And even when works did mention snails, they kept them at higher levels. They're at family and genus. So the Park Service crew thought that there was at least one non-native snail species. They thought there was the Chinese mystery snail. But when there's one invasive species, are there more? And what's their impact on the rest of the ecosystem? The Upper Delaware also has different temperature zones. There's a warm water and a cold water. The cold water segment exists because of a bottom releasing upstream reservoirs that are in the branches of the river. We were curious to see if there would be impacts on temperature for the different communities of snails in these zones. The river and the ecosystem are heavily influenced by anthropogenic activity such as dam releases and recreation. So in order to create effective and responsible river management plans going forward, they wanted to understand the ecosystem of this historic region and to find out what kind of snails are there. So our project goals were to survey the snail community in the Upper Delaware and to form a comprehensive species list of this snail community. We wanted to look at abundance, species richness, and the abiotic influences. We also wanted to see what species were native and introduced. So we sampled for 2 years, in 2022 and in 2023. In 2022, we had 30 different sites that were spaced out every 4 kilometers, the entire Upper Delaware River. Our sites coincided with locations of Atlas eDNA sites. The Park Service project advisors wanted us to utilize these sites in order to compare to future and past work. These sites for 2022 are indicated in the map as the black dots. After our first year, we did notice that there was a gap in certain habitats that we didn't get to sample. So these unique areas weren't included in the survey. So in order to include these in the second year, we sampled 24 sites selected based on that criteria. So these were backwaters, islands, channels, and access points. Those sites are indicated in bright green, and the different shapes show the different types of habitats that we looked at. So each year we would set out in July and August to find our snails. Most of the sites could only be reached by the water, So in 2022, we used kayaks and canoes as our main source of transportation. And the river wasn't always so still and serene as this picture depicts. It got kind of scary when our fully loaded canoes would hit some rapids, and we'd have to make sure everything was hunkered down. But it was still kind of fun. With our GPSs in hand, we would get to our sites and locate where we were going, pull over to the bank, and begin our sampling. In 2023, many of our sites could be reached by car because they were access points that park visitors would go to. The sites that were trickier to reach, like the backwaters or the islands, we would use canoes again. So this is a view of what it would look like going down the beautiful Upper Delaware River. So every site that we went to was different. We would try to be as detailed as we could when describing each habitat by using a habitat assessment. No, sorry. That's for the next slide. Bringing it back. So once we would get to our sites and the boats were all secured and everyone was fastened down, we would collect our water quality data and temperature, and we would lay out a 100-meter transect. Once the transect was laid out, the snails would be collected using a combination of kick nets with D-framed nets and a timed hand picking along the banks of the river within that 100-meter transect that we had laid out. In this picture, you can see what our hand collecting kind of would look like. We'd be searching, looking down on rocks, and everyone would take a different type of microhabitat to collect the snails. And you can see that there are two parks service employees that are in a canoe because this backwater was so mucky, we couldn't actually walk out the transect. We had to row it out. So that's what they're doing. Okay, now every site that we sampled was different. So we tried to be as detailed as we could. Since we had different types of substrates and different types of areas along the banks of the river, we wanted to be super detailed. Here you can see the parks team. They're completing the pebble count, which would assess the substrate types along the whole transect. So looking at what the bottom of the river was like. This is also where we would make note of human impacts, such as buildings, boat launches, or trash, which snails love to hide in. There's a lot of good food in there, I guess. In 2022, we noticed that snails were, particularly the invasive snails, were in these areas of high disturbance and human activity. For example, we would launch from Narrowsburg Access, where we saw many different kinds of snails. But once we got to our site, which is about 50 meters away, there were very few snails around. So we knew that those access points were an important habitat to sample. So the following year, we would sample there. So we utilized genetic tools such as environmental DNA and tissue barcoding of the CO1 mitochondrial gene in order to help us identify to species. These techniques would be helpful in order to observe the diversity of the ecosystem and also identifying, especially when morphological traits for snails were ambiguous. Here you can see Tanya is filtering water for eDNA. Water was filtered at 10 different sites in 2022 that coincided with the eDNA Atlas sites. We used kits from Jonah Ventures to collect and filter the water, which would then be returned for DNA extraction and analysis. Primers for mussels, bacteria, and fish were used. 475 individual snails from 2022 had tissue samples taken and removed, and they were sent off to the University of Guelph, which would then be barcoded, barcoding the CO1 mitochondrial gene. These would then be matched with a type specimen on the BOLD database to inform us what species we had. So after months of rough sorting the samples and IDing the snails with the instruction and guidance from Art Bogan of the North Carolina Museum of Natural Science, we found a lot of snails. In both years combined, we had over 18,000 individuals. And so let's take a look at what our results are. So in 2022, we found over 10,000 snails from 7 families that we were able to identify 13 different genera. We'll look at those all individually in a little bit. So pictured here is a snail. This is a Campyloma decisum. They are a native snail in the family Viviparidae. This map shows the number of families at each site indicated in the size of the circle, and the number of snails found at each site, which is indicated by the color. So the darker the color, the higher the number of individuals. Interestingly, the abundance and the number of families, the diversity, were not always positively correlated. For example, site 49 PA, they had 3 families found, so kind of low diversity, but one of the families, Amnicolidae, had over 700 individuals. Whereas at site 45, they had high diversity. They had 6 families found, and in the family Amnicolidae, they only had 13 individuals. So that was kind of interesting. So our results for eDNA, we found that there were 6 different kinds of bivalves and over 20 species of fish, including the American eel, fall fish, and the eastern Alepio mussels. We even got some mammals, which was cool, like the common shrew. Researchers from the Smithsonian Environmental Research Center, they analyzed our bacteria results for eDNA and found that there were many types of feces in the Delaware, so that's always good to know. This is our tissue barcoding results. It allowed us to see how diverse the snail community was. For instance, the family Physidae. Physids are very difficult to identify morphologically due to the variety in shell shapes. Barcoding allowed us to see how many different types of species of Physid were in the Delaware River. So here's an example of the neighbor joining tree that we have. It shows that there are 5 different species of Physella snail. So the colors of the pictures coordinate to the colors of the on the tree. So you can see that there is some different shell morphology, but none of them were very consistent. Okay. So what's interesting for our results, there were, sorry, everybody, give me a second. For most of our results, there were no species listed in the BOLD database. And that could be due to 2 possible reasons. A type specimen had not yet been barcoded, or the species that we had found, or we may have found a new and undescribed species. But we don't know yet until a taxonomist takes a look at our species to compare them to collections. Okay. Here we go. All right, so here is our species list for 2022. The pie chart shows the family breakdown. So we can see that Physidae and Amnicolidae in that orange and dark teal color were our most commonly found families, making 44% and 39% respectively. While Valvatidae, which was our least commonly found snail, we only had 1 individual from them. Snails are generally hard to ID to species based on their physical characteristics. Many times you need to look at their genitalia or mouth parts, which is hard when you save them in ethanol and they kind of break down. So we were just looking at their shell morphology. And based on those traditional methods, we are able to identify 7 families and 9 species. Those are indicated in this middle section. With our tissue barcoding, however, we were able to delineate to 27 different species. 16 of those species are in that barcoding limbo where there either is not a type specimen on the BOLD database or they're a new undescribed species. But we have to wait for taxonomical review for that. 3 families that were difficult for me to delineate were the Physid family, which you can see there were 7 species that came from the one species, that came from the one. So they were able to delineate 7 with the barcoding. Enselinidae, which are actually limpets, which is really cool. I didn't know they were freshwater limpets. That breaks down into 4 different species with barcoding. And then Galba. Galba is a limnaeidae, and they're a genus that is difficult to identify, but we were able to pull out one mystery species, and then also a species, Galba humilis. So here's just some looking at some of the diversity under the dissecting scope. So this is what I would see when I look at a tray of the snails. This first picture is of 2 different families. So the one on the left is a Ficella, and the one on the right is a Galba. Physids were the only left-handed snail that we found in the Delaware, and both groups were very difficult to delineate. It's hard enough to delineate them themselves. So it was interesting to see that these groups are very small and all similar looking. So the middle picture are the Amnicolidae. They ranged in size from about a pea to the tip of a pencil. So they were very small, and they would come in large quantities most of the time. We discovered, based on our genetic results, that actually we had the species Amnicola limnosus, but we discovered that there was a misidentification, and there was actually a whole other family, Lithoglyphidae, and with the genus Somatogyrus within that family. And finally, this individual is from the family Plenorbidae, and it is the genus Gyralis, but we nicknamed them in the field cinnamon rolls because they had that perfect little coil, and so we'd always get really excited when we'd find one. So now let's look at the results for 2023. Now, we were looking again at specific habitats, so this was river accesses, backwaters, channels, and islands. In total, we found over 8,000 snails in 7 families and 12 genera. This is a picture of a mystery snail with, I don't know if you can see it, but there's another snail on top of it, and that is a Pseudosynchinae, and that is not staged. That's how we found it. It was pretty silly. This map shows, again, the diversity and abundance of our 2023 data. The darker colors indicate a higher number of snails, and the circles correspond to the amount of families. The site with the highest diversity was site New York 23. They had 6 families. This was also the only site where we found Valvatidae in 2023. The greatest abundance was a backwater, PA 27, which 74% of those snails were from the family Physidae. And then Mylanville access, PA 15, had a high abundance and diversity. This is also the northernmost site where we found the mystery snails. So here's our taxa list, again for 2023. The pie chart shows that Physidae were overall, the orange space, the largest and most common snails found, making up 58%. Some differences from the year prior, Pleuriseridae and Amnicolidae both had decreased, and all other families, however, had increased both in percent and in number of individuals that we found. This year, the taxa list for this year looks a little bit different. The areas that are in gray don't pertain to this year because we weren't able to do any barcoding. So species that would have delineated, such as Ficella, we weren't able to be as detailed with the list this year. Also, one species that we did not find at all in 2023 was Campyloma decisum. So they are also not on the list. So here is just another shot of some of the amazing diversity that we found with the snails. Here are actually 6 different families. So I will start from the first third of this page. So the first snail is a Physid snail. They were the most abundant family that we found, And then next to that, we have a Lithoglyphidae, that family, the genus Somatogyrus, which looks very similar to Amnicolidae. And then below that is the Valvatidae. This is a Valvata bicarnitata. They were present in larger numbers in 2023 than in 2022. Remember, this was the individual that we only found one in 2022. They have this really cool ridge that goes all the way around on both sides, and it really sticks out. That's called the carnita, so hence their name. In the middle here, one on top of the other, they look very similar. This is a Galba humilis, and then underneath that is a Pseudosynchinae columella. The Pseudosynchinae, if you can see, it has a much larger exaggerated aperture, which is the shell opening. So the tall one with the white background, that is a Pleuriseridae. That is the species Elamia virginica. Before we knew what they were, we called them the Statue of Liberty snails because the adults would get really kind of mangled and they'd get messed up, but they would have this greenish, bluish color all over them. And so they kind of looked like the color of a Statue of Liberty. So that was our fun little thing. Next to the tall, on top of the tall one is a limpet. It looks like a little hat, a little dome. Limpets were actually the only group within the Planorbidea family that in 2023 decreased, which is very interesting. Next to the limpet is a Heliosoma anceps. This also has that kind of white looking ridge around it. They have that on both sides, and that's a distinguishing key factor because the one below it gets mistaken for them all the time. This is a Planarbella trivolvus. On this side, from this view, you can see that it's very smooth and kind of almost like bubbly, but on the other side it has this bowl shape to it, which is similar to the Heliosoma anceps that is on top. So a major environmental factor that we looked at was temperature. While the Delaware River temperature gradient is impacted by tributaries and surrounding reservoirs and power plants, we found that the largest impact was actually the Cannonsville Reservoir. The Cannonsville Reservoir releases cold water from the bottom, which then changes the temperature downstream, thus creating consistent temperature zones along the Upper Delaware. The inlaid map shows the Cannonsville Reservoir just north of our park. The river transitions to warmer, more natural flowing temperatures around Callicoon, which on the map is indicated with that red bar. We did find that there's a difference in temperature upstream of Callicoon and downstream of Callicoon, with a significant p-value near 0. This map shows the catch per unit effort for our that we calculated for each site. We took the total number of snails that we would find and then divide it by the amount that we spent sampling. So this map is specifically showing our hand sampling data, and usually that was over a 30-minute time period. There's a positive correlation between the catch per unit effort and the temperature, and we can see that the transition from cold to warm and the increased catch per unit effort are in the same area. So you can see between the top part of the river and the lower part, there is a huge difference. It's pretty plain to see there. The highest catch per unit effort in 2022, actually and overall, was Site 57 PA. They had over just under 1,200 snails at that site with a catch per unit effort of 398 individuals in 30 minutes. This is the largest dot kind of towards the end of the map. And then the highest rate for 2023 was at site PA 27, which is the largest triangle kind of at the end of this map, where they had, their catch per unit effort was just around 280 individuals in 30 minutes. So why is this important? It's interesting to consider, especially when you look at the impact of more frequent dam releases, and that could possibly change this water gradient. And it could definitely impact the snail community that is further downstream. As we think about climate change as well, and other unpredicted changes that can come, it's really important to understand and be able to forecast the impacts on the snail community. It's important to consider the impacts of changing temperature gradient and climate change on invasive species as well. So let's talk about these mystery snails. They are an invasive, non-native snail originating from Eastern Asia. There's much that we don't know about them that's still being studied and learned. For instance, their impact on native snails is still indeterminate. We found mystery snails at 7 of our 54 sites, which is smaller than what we thought, which is a good thing. But we did confirm that instead of the Chinese mystery snail, which is what we thought we had, we actually have the Japanese mystery snail, Cepengupaludina japonica. And we found this based on the tissue barcoding of the CO1 gene. So mystery snails are clearly very difficult to tell apart. So we had to use genetic tissue barcoding in order to determine which species that we had. So here is pictured a snail that we actually barcoded. Its name is snubbed 40223. And you can see the neighbor joining tree that we had 5 successful matches with the BOLD database. One of our contacts that we have made in just this discovery of mystery snails are actually doing, if you can send them your sample of mystery snail. So I have these handouts up front here, but you can send in your mystery snail if you have found one, and they will do a genetic analysis and tell you what kind of snail you have, because it's very difficult to tell apart. So if at the end of my talk you can come up and grab one, and it's just a great resource that we discovered. So just wanted to have a little plug in there for them. Okay, so looking back at our mystery snails, in 2022 we had 48 at 3 sites, and in 2023 we had 348 at 4 sites. Most of the sites that we found them at were access points, which supports our hypothesis that we would find more non-native snails in disturbed locations, such as access points. Narrowsburg, what I had talked about before, actually their catch per unit effort for mystery snails alone, was 83 individuals in 30 minutes. So it was pretty full of mystery snails. So we also had 2 sightings of mystery snails in this in-between spot on the map that we didn't find any in 2024 at 10 Mile River and Lackawaxen Access Point. The Japanese mystery snails seem to prefer warmer water as they are below Callicoon. However, it's interesting they're not further down the river where at similar sites. So we don't know why yet that's the case, but we have been considering that it's an issue of suitable habitat. So here is, these are different ages of the mystery snails to see what they kind of look like. They would range in size from huge to then very small, and they look very different. They're very angular in a juvenile form, and then they become more bulbous and giant. So the impacts of mystery snails is generally unknown. This is still something that is being actively researched. However, they have been in North America for over 100 years. While there's been more research on the Chinese mystery snails, even their ecology is still, and impacts are still unknown. In a study by Schuler et al., they found that the introduced species in the same family, Viviparidae, the banded mystery snails, had a significant impact on nutrient cycling with nitrogen and phosphorus levels. While this study did not observe Japanese mystery snails, it begs the question, do they have a similar impact? A future pilot study by the Academy of Natural Sciences will be investigating impacts of mystery snails in urban parks, and specifically the impacts on nutrient cycling. So with all of our projects being done and all the sorting of the snails, we deposited our snail samples into the malacology collections at the Academy of Natural Sciences, where our snails will be cataloged and integrated into the collection. So the Park Service and the Academy recently received a grant from the National Parks Foundation to implement a mystery snail removal and monitoring effort. So we hope to test different removal methods, such as hand sampling, but also using traps to track their effectiveness over time. Fieldwork will begin this summer, and we are very excited to work with the Park Service, with volunteers, and with the community to protect and preserve this amazing river ecosystem. So we would just like to thank all of the people who helped us with this project. We appreciate the support, their time to get this work done. This project was made possible through the support of the National Park Service Inventory and Monitoring Division. Here are some of our references, and I think it's time to open it up for questions and discussion.
I know you said there's not a lot known about the mystery snails, but so you said you found a lot of the access points. Could that be because people aren't like washing their boats off if they're taking them in and out of the river? Is that probably like a big reason why they're
we
there?
think actually why they're there is because people get them through aquatic trade and they just dump their aquariums. So but then they are so small, so it's easy to transport them on a boat or on waders. I even found, after field work, would find them in my shoes, like all snails in my shoes. But so it was crazy. So it's like, wow, I didn't realize that they could be transported so easily.
Wow. Okay, I didn't think like people were like dumping like their. I mean, I guess
it's a huge
fish
issue.
all the time, so
And it just really takes one, and then they just become, they're really prolific. So, thanks. Good question.
Just sitting out there waiting.
That'd be awesome. Okay. Nick?
Hi. So were only Chinese mystery snails found, or were only Japanese mystery snails found throughout the study, or were there any Chinese mystery snails? And are you aware of any other populations outside of the Delaware where there's Chinese mystery snail, or do you think this may be a stronger population of Japanese even outside of Delaware?
Yeah, well, it's brought up the question of, do we even know if the Chinese mystery snails that people have found before are Chinese, or are they the Japanese variety? So actually our friends at the George Mason College, that was their issue too. They were like, we don't know what we're finding. Is it a Japanese mystery snail? Is it a Chinese mystery snail? And what implications and what differences are there between those two? In our research, we found what we think are only Japanese mystery snails. Out of the, I think, like over almost 400 that we found, we think they're all the same. Based on their morphological traits, we were able to kind of compare, and none of them kind of looked extremely different. So we can confidently say right now that based on our genetic data and just the samples that we have, that it's Japanese mystery snails. But there are ponds and lakes and reservoirs all over near us and in the watershed that have Chinese mystery snails as well. So, yeah.
Kind of jumping back to what I was talking about with the washing off like your boats or your waders or your shoes or dump your aquarium. So for like potential community outreach, could you like make signs? I don't know if you guys are like maybe thinking about doing this and like putting them up like where you're finding them. Like, hey, like aquatic hitchhikers, like make sure like you're finding these snails, and maybe you could maybe even reference like how many you're finding there. You have like certain access points where there is more, like, oh, we're finding this many snails and it's potentially because of this, so let's all do our part. I don't know.
Yeah, for sure. I think that's one of the things we hope to do is with this upcoming work is to kind of test out different methods of, does hand collecting and just removing them actually help? Does it clear out the areas? We really want the community to be engaged. It's like we're visitors to the river, and so the people who actually live there and care about it, we want to engage them and to just tell them what's going on, because half the time, like, people don't really know that there are so many snails. It's like, I didn't know. I was like, as an ecologist, this was kind of mind-blowing. But yeah, we hope to engage the community, and it's an exciting prospect actually to have, so. Thanks.
In regards to the amnicolidae and amnicolidae-like snails, did you say, was there a record of, I'm going to say this wrong, Lithogliphidae, or were there representatives of that family found, or was that just a comparison between the two that they're?
So it's not out of the question to have found the Lithoglyphidae. Yeah, it was possible to find them. To me, they were so similar, almost to like cryptic level, where you would have like a whole tray of them, and it's like they all look the same. They all look like Amnicola, and it was until we found through our tissue barcoding that it was like, no, there are actually two different groups here. That it was like, okay, let's look at these. To me, then after looking at that, it's like, okay, there's one morphological difference, and it's the difference in the like body of the snail. There's a vein that you can see, and that's the only difference that I could find. Now, Amnicola also had the vein, but it was like a different shape. So it was like the lambda shape versus like just a line. So that was kind of the difference, and I was like, I don't know if I can say as an ecologist, not a taxonomist, that this is for sure a key point different, like a keying difference. But we hope that when the taxonomists at the Academy review them, they can make that decision. So,
okay,
yeah.
so we do have that family here in Delaware.
Yes.
That's great.
Yeah, I know. The only non-native that we found was the Japanese mystery snail, which was amazing. So, especially with the concern of there's a New Zealand mud snail that's very small, and thankfully we did not find that. So, it's good news. Go biodiversity. So, thank you all. Any other questions?
So all those native ones and everything that you took out, you never put anything back? You took them?
Nope.
Okay. I just was wondering.
Yeah.
I kind of thought because you're taking things out.
I know. It's one of those things where it's like you have to kill what you love to learn, but clearly, I mean, the amount that we took out, there were still so many throughout the location. So I don't feel too bad in the name of science, but
I'm assuming none of them are endangered or
No.
threatened or
Nope.
anything. Yeah.
yeah. So very cool. Thank you all.
Thank you so much.